Hello, everybody. My name is Uli Funken, and on behalf of the Peninsula Parkinson's Research Interest Group, or PenPRIG, I'd like to welcome you to this Meet the Researcher Online Hour. This is our third such meeting after we had Claire Bale of Parkinson's UK in May and Will Young of Exeter University in June. Today, I'm very excited to welcome Simon Stott, who is the Deputy Director of Research at the Cure Parkinson's Trust. You may also know Simon as the author of the weblog, The Science of Parkinson's, which is Simon's very personal crusade to break down the language barrier between research and us mortals. Simon will talk to us on the topic clinical research focused on disease modification, which is also known as finding the magic bullet. Participating in the session are a mixture of PenPRIG members and supporters who will be asking some questions after the talk. With that, I will hand over to Simon for his presentation.
So thank you very much to the organisers for the invitation to speak today. I'm just going to share my screen and we shall get started. So today I'm going to talk about clinical research that's focused on disease modification for Parkinson's. And the first obvious question to ask is, what do we mean by disease modification in Parkinson's? And that, of course, leads to another question, which is, what exactly is Parkinson's? So I'm going to start off by briefly giving you an explanation of both of these concepts, and then we will look at 5 of the clinical trial programs that are ongoing at the moment, which we should be keeping a close eye on. Now, Parkinson's is classically considered the second most common neurodegenerative condition after Alzheimer's, and it's characterized by a triad of motor features: slowness of movement, rigidity of movement, and arresting tremor. But there's also a large assortment of non-motor features as well. For example, sleep disruption, loss of sense of smell, and also there can be gastrointestinal issues, as just to name a few. The motor features, however, are the most predominant aspect of the condition. And this is characterized or correlated with the loss of dopamine neurons in the brain. And dopamine neurons, you can actually visualize them with your eye. If you look at a section of the human brain at the level of the brainstem, so sort of down towards the bottom of the brain, if you cut through there, preferably after people have passed away, you will be able to see the dopamine neurons because they have a black pigmentation. And so here you can see an unaffected individual's brain, someone without Parkinson's, on the right-hand side. And I'm not sure if yes, you can see my cursors, hopefully. You can see there are 2 dark pigmented areas on both sides of the brain. And this is called the substantia nigra. And the dark pigmentation is called neuromelanin. Dopamine neurons produce neuromelanin, and as a result, you can see them visually. This is black pigmentation. And what you notice when you compare the right to the left-hand side is from a brain of a person who passed away with Parkinson's, and they have a lot less of this dark pigmentation in the substantia nigra. Now, the dopamine neurons down here in the brainstem, they send branches or axons, they send branches or axons up into the higher regions of the brain to 2 areas called the caudate and the putamen. And this is where the bulk of the dopamine in your brain is actually released. Gradually, with Parkinson's, we see a loss of these branches, or as they're called, axons, as the dark pigmented neurons are being lost. So we get less and less of the branches going up to the putamen and the caudate and releasing dopamine. And we can actually see this in the hospital. You can have what's called a DAT scan. And the person on this is a bird's eye view, so looking down on the brain of both a person on the left who is unaffected by Parkinson's and a person on the right who has Parkinson's. And what you can see is the amount of dopamine turnover here on either side of the brain in the putamen. And on the left-hand side, the unaffected brain, you can see there's a lot of this white and orangey red coloration. And there's a lot less of it when you look at the person with Parkinson's. So there's a lot less dopamine turnover. So that's what, that's a very brief summary as to what Parkinson's is. the normal trajectory with Parkinson's is that there is some indication of something not quite right. And then the individual will go along and get a diagnosis from a neurologist. And that will lead to them being diagnosed with Parkinson's and the initiation of symptomatic treatments. Now, Parkinson's is a progressive condition. As a result, it's a downward sort of trajectory. Slowly but surely, they're losing more dopamine neurons and the symptoms get progressively worse. The initiation of symptomatic treatment certainly helps in improving quality of life, but it doesn't actually alter the trajectory. The trajectory is still continuing to get worse. So when we talk about disease modification, we're talking about any medical treatment of a disease that changes the trajectory of that condition. So we're looking for a therapy that will slow, stop, or reverse the disease. If we go back to that schematic, we're looking for a treatment that we can give a diagnosis, or ideally before the motor symptoms even appear, and return either slow down that progressive deterioration, or return the individual to some sort of level of baseline. So that's what we mean by disease modification. And when we talk about clinical research that's been conducted on Parkinson's, there's quite a lot going on at the moment. And I'd point your attention at this point to, don't click on that, otherwise all sorts of weird things will happen, yeah. I'll point your attention to a recent review article where we have got 3 Parkinson's research advocates who, Sue Buff, Gary Rafaloff, and Kevin McFarthing, who took all the data that's currently available on the clinicaltrials.gov website for clinical trials that were conducted during 2019 through to 2020. And they conducted an analysis and statistics on this. And they've broken them down into different types of treatments, etc. And what's really remarkable, if I can show you this here, is that they found there were 145 clinical trials for drugs focused on Parkinson's. And 57 of those, so almost 40% of them, are focused on disease modification. Now, one important detail here is that there are 3 phases of clinical trials for clinical development, excuse me. Phase 1 is where you test your drug in humans. It's a very short phase. And basically, the goal is to check whether your drug kills someone or kills people or not. If it's safe and well tolerated, then you go to phase 2. And that's where you're testing the safety and tolerability of the drug in your population of interest. So in our case, Parkinson's. And you're also looking for signs that it's doing what it's supposed to do. In this case, with disease modification, we're looking for any evidence that could be slowing down the progressive nature of the condition. And then in phase 3, you're shifting your attention to long-term safety and tolerability in a very large population of your cohort of interest. And the ultimate test at this level is whether the drug is actually doing what it's supposed to do. And so here we have, you can see at phase 3 at the moment, we have 3 trials for disease modification. And one of them we're going to talk about very shortly is this drug called Exenatide. Now, there are 5 clinical trials that I've sort of chosen today just to discuss and that are of interest for the Parkinson's community, I think. And these are 5 that we should be keeping an eye on. There's a lot of other clinical trials ongoing, so you shouldn't limit yourself to just these 5, but I find these 5 particularly interesting. And the first one, as I was saying, is the Exenatide trial. So Exenatide is a diabetes drug that's being repurposed for Parkinson's. Exenatide is used in diabetes to regulate the levels of insulin and glucose in the blood. And back in the sort of late 2000s, researchers started noticing that Exenatide also had beneficial properties in models of neurodegenerative conditions. Drugs seem to be neuroprotective in models of Parkinson's. And they researched this further and further, and then eventually they conducted a phase 1 clinical trial. It was an open label study in people with Parkinson's. And that led to the results were encouraging enough there that they went on to do a phase 2 clinical trial. And the results of that phase 2 clinical trial were published in 2017. Now, what you can see here is that the study involved 60 people, and they were split into 2 groups. One group was treated with a placebo, so an inert control substance, for 48 weeks. And those individuals, as you can see, gradually got worse with regards to their motor symptoms. But in the Exenatide group, so the blue line here, those individuals who were treated with Exenatide for 48 weeks basically got better and plateaued at that level until at 48 weeks the treatment was taken away, and then gradually they started to return back towards their baseline level. So this was a very encouraging result. What should be noted is that the positive result was only seen in the motor symptoms, but it's still a very encouraging result. It gave us our first sort of signs that we could potentially be modifying the disease. And that was led by Professor Tom Fultonay here, who is at UCL in London. This is a different Tom Isaacs. But it's led to the initiation of the phase 3 clinical trial, which is ongoing at the moment. And this study is recruiting 200 individuals who will be assessed for 2 years with either Exenatide or placebo treatment. And that's not the only study being explored with regards to Exenatide. Exenatide is what you call a GLP-1 agonist. And there is a large selection of GLP-1 agonists available. And Liraglutide is another one. And that's being clinically tested at the moment in Los Angeles in individuals who are insulin intolerant. So this is a sort of a diabetic population that has Parkinson's. We're hopefully going to see the results of that study in 2021. And there's also Exenatide. This is another GLP-1 agonist, very similar drug to Exenatide, which is in a phase 2 clinical trial in France. And we will also hopefully be seeing the results of that study in 2021 as well. So there's a large number of GLP-1 agonist trials going on at the moment. And we should be having a lot more information with regards to whether this class of drugs is having an effect over the next couple of years. So that's one to watch out for. Another drug to look out for is Ambroxol. This drug is a respiratory medication. It removes mucus and inflammation in the throat and lungs. It's widely used in Europe, but not so much here in the UK or in the US. But a few years ago, researchers noted that Ambroxol was raising levels of an enzyme in cells called glucocerebrosidase, or GCase for short. And this enzyme is involved with the waste disposal system of the cell. It's an enzyme that helps to chew up and dispose of unwanted rubbish in cells. And what they noted was that this drug called Ambroxol was having neuroprotective effects in models of Parkinson's. So that led to a phase 2 clinical trial being set up and conducted by this gentleman here. This is Professor Anthony Shapiro, who's working at the UCL in London. And that study was called AIM-PD, which was Ambroxol and disease modification in Parkinson's. It recruited 18 individuals, and they were treated for 6 months. And I want to point out here that these individuals were heroic, because not only were they required to take 21 pills a day on top of the Parkinson's medication that they were already taking, but those 21 pills are the most bitter pills that you could possibly imagine. So their involvement here was absolutely heroic, and it wasn't it wasn't without benefit. As you can see here, the drug was well tolerated. Excuse me, when they looked at the results. It didn't have adverse effects in the body beyond the actual taste of it. But it also raised levels of GCase in the brain. In the graph you can see here below, you have individuals who have what's a GBA mutation. GBA is the gene that makes, or the region of DNA that makes GCase. And it is also a genetic risk factor for Parkinson's. If you have a GBA genetic variation, you are at a higher risk of developing Parkinson's than someone without. And your Parkinson's is more progressive than individuals with Parkinson's who don't have a GBA mutation. So we had a combination of both people with and without GBA genetic variants in the study. And you can see that across both groups, there was an elevation in the levels of GCase in the brain. So this was very exciting, and we thought this is obviously worth further investigation, and the next stages of this clinical trial program are now under development. And if you'd like to be part of that clinical trial program, that particular one, you can have a look at this website called PD Frontline. Don't bother writing all the information down. I'm happy to make the slides available. But this will be, this study here is trying to get a trial-ready cohort for the next step in the Ambroxol study. We're just currently working on a reformulation of the drug. But Ambroxol is not the only clinical trial that's focused on this GBA pathway. There's also a biotech company called Prevail in the US. And Prevail are focused right now on a phase 1 clinical trial that's looking at GBA-associated Parkinson's. They're taking what's called a gene therapy approach. And gene therapy is a method of treating people with DNA rather than drugs. So they're going in and they're actually correcting the GBA gene, the region of DNA that gives rise to this protein, GCase. So they're going in and correcting it, or they're going in and inserting a new version of it. And hopefully that treatment will be found to be safe and effective. We shall see. Another one to keep an eye on. Third on my list of 5 is prasinezumab. I can never say correctly, prasinezumab. This is an immunotherapy approach. So instead of boosting the waste disposal system of the cell, as we were with Ambroxol, in this case, we're boosting the immune system. Parkinson's is associated with the buildup of a particular protein in the brain. This protein is called alpha-synuclein. It sounds like a distant galaxy, but it's actually one of the most common proteins in a neuron. It makes up about 1% of the proteins in the neuron. And what you find in Parkinson's is that it starts to cluster together, clump together, and form these little aggregates that we call Lewy bodies. And one theory of Parkinson's is that the progression of Parkinson's could be associated with the passing on of a toxic form of alpha-synuclein from one cell to another. And so the immunotherapy approach is to design these small Y-shaped structures you can see on the picture here, these small Y-shaped proteins. These are called antibodies. And our immune system uses antibodies to label things that it doesn't like. So what researchers have done is they've designed antibodies that can actually target this toxic form of alpha-synuclein and bind to it as it's being passed from one cell to another. And then the immune cells can come in and clear it away. And one clinical trial that has garnered a lot of attention recently is the prasinezumab study. It's a phase 2 clinical trial. The study is called the PASADENA study. And it's been conducted by Prothena, the small biotech company, and the large pharmaceutical company Roche. And in April, the companies announced that the study had not met its primary endpoint. So the primary endpoint is the predetermined measure of whether a trial is successful or not. And they said that it hadn't met its primary endpoint. But they said that the treatment did show signs of efficacy. And just recently, a couple of weeks ago, they made a presentation where they presented these graphs here. Now the graph on the left is the primary endpoint. The primary endpoint in this study was using the Movement Disorder Society Unified Parkinson's Disease Rating Scale, or UPDRS, total score. So this involves 3 separate parts, parts 1, part 2, part 3, combined. And as you can see, the placebo treatment group, this is in blue, the blue line, is very similar to the 2 doses, a high dose and a low dose of the prasinezumab. And there's not much separation between them. So this suggests that there's no difference between the 2 groups, or the 3 groups, with regards to efficacy in terms of UPDRS score. But when you break the UPDRS down into separate components and you focus just on the movement component, so these are the movements, the motor symptoms aspects of the assessment, what you see is a separation of the data. So the placebo group continued to progressively get worse here, but the red line, which is a combination of the low dose and the high dose of the prasinezumab, they sort of immediately after baseline start on a lower trajectory and gradually climb to a point, and then all of a sudden, as the company is suggesting, they plateau, and this is the green circular area here, they plateau across the last stages of the study. So the companies are both very excited about this result, and they are now exploring what to do next with regards to this clinical trial program. And we'll hopefully see the full results of this study. Thus far, we've only had press releases and some oral presentations, but we'll hopefully see the full set of study results in late 2021. And the PASADENA study is not the only immunotherapy approach being attempted with Parkinson's. There's also another phase 2 study from the pharmaceutical company Bydureon. That's involving 300 people, and we'll hopefully see the results of that in 2022, 2023. So that's another one to keep an eye on. And a third immunotherapy study that we should also keep an eye on is from a biotech company in Austria. This company is called Apheris, and they're developing, rather than designing antibodies, what they're actually developing is a vaccine. So with regards to the Roche and the Biogen approaches, they are designing an antibody in the lab, and then they're injecting the antibody into people with Parkinson's. Apheris is actually they've actually developed a vaccine for Parkinson's. So this is a small fragment of the toxic form of alpha-synuclein. They're injecting it into people. This is causing the immune system to react. And as a result, the immune system not only targets the vaccine, but also the toxic form of alpha-synuclein in the brain and the rest of the body. Now they've just finished phase 1 research on their vaccine, and they're looking to start phase 2 later this year, although there's not much of 2020 left now. But they're, hopefully, COVID willing, they'll be starting very shortly with the next stage of this clinical trial development. So that's immunotherapy. That's a very interesting area to keep an eye on. The fourth clinical trial of interest for the Parkinson's community is UDCA. UDCA is a bile acid treatment. It is used for breaking down gallstones and treating liver conditions. But a group in Sheffield, sorry, this is it here. A group in Sheffield observed that UDCA was also having neuroprotective benefits in models of Parkinson's, particularly around the idea of the mitochondria. Now, mitochondria are the power stations of a cell. They give the cell all the energy that a cell needs. And the UDCA was enhancing or making the mitochondria function better and improving the activity of mitochondria, not only in classical models of Parkinson's, but also in genetic models of Parkinson's. So this led to a 48-week study of UDCA in people with Parkinson's. It's a phase 2 clinical trial that's involving 30 people diagnosed with Parkinson's. And it's being conducted by Professor Oliver Bandman in Sheffield. And Heather Mortiboys was the researcher who did a lot of the preclinical studies on UDCA. And we'll hopefully see the results of that UDCA clinical trial in 2021. There's a lot to keep an eye out for in the new year. And just recently, UDCA has been in the news. A company called Amylyx in Boston, they have a dual therapy, which is involving a form of UDCA, and it was trialed in 137 people with ALS, so motor neurons disease. And what they found was it significantly improved the function in the level of function in the individuals with ALS. And so this is giving further support for the idea that UDCA potentially is has neuroprotective or beneficial effects in the brain, not just with regards to gallstones. And this involves TUDCA, which is a different form of UDCA, I should add, but it's still TUDCA gets broken down into UDCA. So that's exciting stuff. And one fifth clinical trial to keep an eye on is a clinical trial that's being conducted by a company called Denali, and it's focused on LRRK2 inhibition. The gentleman in this picture is Sergey Brin. He's the co-founder of a small company you might have heard of called Google. And his family is affected by Parkinson's. He and his mother and his auntie all carry a genetic variation in a region of DNA called LRRK2. And this increases your risk of developing Parkinson's at a younger age. And he's been channeling a lot of money into, he doesn't show any signs of Parkinson's yet, and he might not go on to develop Parkinson's, but he's certainly not taking any risks. And he's been channeling a lot of funds into the development of research regarding LRRK2 and Parkinson's. And that's led biotech companies such as Denali to focus on inhibiting LRRK2 in people with Parkinson's. So when you have this genetic variant in the LRRK2 region of DNA, or the gene LRRK2, it results in a hyperactive form of the protein. And in a very carefully balanced environment like a cell, you can appreciate that a hyperactive protein is not a good thing. And so Denali, in collaboration, just recently, they've announced a collaboration with Bydureon, a big pharmaceutical company, and they are taking forward a drug, DNL201, which is a LRRK2 inhibitor. It tones down the hyperactivity of the LRRK2 protein. And they had successful phase 1 clinical trials, and they'll be hopefully starting the next phase of that clinical development in late 2021. And they're not the only companies looking at the LRRK2 inhibition for Parkinson's as a treatment. GSK, Cerevel, and Bydureon, three other companies that are all currently developing LRRK2 inhibition inhibitors. So one question I get often asked by audiences is, how can I get involved? And it usually follows up with the statement, I'm a little bit nervous about actually doing a clinical trial, so what else could I possibly do? There's a large number of different roles people in the Parkinson's community can take, from simply communicating and advocating for Parkinson's research, through to mentoring people and being there to support those who are going through the clinical trial process. So there's many, many different ways. And I implore and encourage everybody watching this to actually get involved. It's a very exciting time for Parkinson's research. there is the opportunity for us to really make a big difference. There's certainly a huge amount of research going on in a vast number of different ways. We're attacking this condition. And at this point, I'll leave you with a quote from one of my heroes. He's a fellow Kiwi, Mike Lloyd. Mike was diagnosed with Parkinson's 10 years ago. And he just recently, last year, in 2019, he ran his 10th New York Marathon. And that's rather a remarkable achievement for somebody who's 10 years into a diagnosis of Parkinson's. And for Mike, it's a celebration of what he can do. He's not interested in what he can't do. He just focuses on what he can do. And the one detail I haven't told you about Mike is that he's also blind. So Mike, he never focuses on what he can't do. He only focuses on what he can do. And people ask him, what will you do when the Parkinson's doesn't let you run anymore? And Mike just says, oh, I'll find something else to do. It's not what you can do, it's what you can't. It's not what you can't do, it's only what you can do that we should focus on. And with that, folks, I'll say thank you very much, and I'll be happy to take any questions.
Yes, thanks very much, Simon. Very exciting and a lot of information you've given us there. So yeah, we come to a Q&A now. There'll be some pre-submitted questions that we've, I think you're aware of, but hopefully there is some time for some ad hoc questions. I definitely want to find out about the hacker role that was on one of the slides there. No, question number one. I know you mentioned 5 areas in your presentation. If you had to pick one of them that would promise you expect the most significant chance for success, which one would it be?
So I'm going to jump back to my presentation here. This is a question I get quite often, and I'll just share a couple of slides with you. So as I was saying, I get this question quite a bit. Hey Simon, which of these drugs do you expect to work? And I have 2 answers to the question. And I warn you now before we start that the second answer is going to disappoint you. But I will explain myself. The first answer is it depends on what type of Parkinson's each individual has. For example, if I had a GBA genetic mutation, I would be focusing my attention on a lot of the GBA approaches, and there are a lot. It's not just simply Ambroxol or Prevail. There are other companies and researchers focused on improving GBA activity. Similarly, with LRRK2 and other approaches. But number 2, I don't expect any of these treatments to work. And before you stop watching or listening, I need to explain myself here. We need to go into this process in an unbiased fashion. Clinical trials, we can't go into clinical trials if we have expectations. So if you go into a clinical trial with the expectation that the drug you're going to take is fantastic, there is the possibility of having a placebo response. And so, and also, I think it's very dangerous to go into any situation with expectations, because the higher your expectations, the more you are hoping for a positive outcome, the higher the chance you'll be disappointed, even if it's a 50-50 chance of a trial or a drug failing. So what I would say is, I try to put across a philosophy of low expectations, I've lowered my personal expectations to 0. I'm a cup is completely empty kind of guy. And the sun came up today. It was amazing. It was completely unexpected. But that, and others in the community also are trying to put across this this sort of philosophy as well. Martin Taylor, who was diagnosed at 32 with young onset Parkinson's, he set up the Parkinson's Research Interest Group on Facebook, and he pushes a philosophy of positive realism. Nothing that we've thrown at Parkinson's has yet slowed or stopped the condition, so we have to be realistic. But there is so much research going on at the moment, there is reason for optimism. That obviously raises the next question, which is, what are the reasons for optimism? And it's just simply the level of research. It's just an incredible amount. From a researcher standpoint, it is impossible to keep up at the moment. And the research isn't just simply focused on proteins like GBA or LRRK2 or alpha-synuclein. There is just an absolute tidal wave of research. Every single day, there is at least 20 or 30 new papers, research papers coming out, and that number is only going up exponentially. This graph shows you the last 200 years since James Parkinson first described Parkinson's in 1817. And across the bottom here are the number of research papers that have been published with the keyword Parkinson's. And to date, this actually is this old number is a bit old now, but it's over 140,000 papers focused on with the keyword Parkinson's during that period of time. And almost 110,000 of them were published in just the last 20 years. We've never had so many research papers. We've never had so many clinical trials focused on Parkinson's. So I think there is certainly reasons for optimism, but we shouldn't have any expectations as to what's going to work or not work.
Right, thanks very much, Simon. I think the next question came from Bob.
Yeah, it did. My interest is around the gene therapy, and we want to know, have they isolated a faulty gene? And if so, how or what is the way forward?
I'm going to share my screen again. I feel like a 1980s TV chef, celebrity chef who says, here's one I prepared earlier. So in 2018, this research paper was published. And what you see across the top, about 50 odd genes. Across the bottom are all of the chromosomes that humans have. And the dots represent individual mutations. So each of these genes across the top, each of these regions of DNA are associated with an increased risk of developing Parkinson's. And you got here such as alpha-synuclein, which has the code SNCA, alpha-synuclein. GBA is right up at the front here. And LRRK2 should be down the back. There it is there. So some of these are familiar to us, but there's about 50 odd genes here that are associated with Parkinson's. And this was in 2018. Last year, this list was updated, and it's now at around 95 genes associated with Parkinson's. Now, the complicating factor here is that there are a lot of different genes associated with Parkinson's, and it's giving rise to the idea that we're not dealing with a singular condition, but rather what's called a syndrome. So it's a syndrome is a group of medical conditions that have similar symptoms, but they have different mechanisms underlying them. And what we might be dealing with is, instead of Parkinson's disease, it could be Parkinson's diseases. And a lot of these genes could be playing a role in various aspects of those diseases. So it's a very, very complicated picture. We can't say that there's a single gene or a single faulty bit of DNA that's causing it. But what's interesting with regards to the biology of these genes is that it's pointing us towards areas of cellular function, such as waste disposal. A lot of these genes are involved with waste disposal. So if you disrupt waste disposal at any stage, you get this set of symptoms that looks like Parkinson's. And then there are other aspects of biological function as well, such as transmission between cells. Some of these genes are involved with that as well. So it gives while there's no one faulty gene, what all of this research here does, it points us towards aspects of biology that we could be looking at. And it's only through this sort of research that we have found genes like LRRK2. We now have clinical trials focused on LRRK2. So I hope that answers your question.
Thank you.
Ian had the next question.
Good afternoon, Simon. I'm going to change the tack a little bit now, because obviously money is very important to you. I've noticed that Macmillan Cancer Support has had a major campaign recently to ensure money keeps flowing into the cancer research coffers, despite the current situation. Looking on your website, I noticed some wonderful efforts at fundraising, but what has been the effect of COVID-19 on Cure Parkinson's Trust income?
So it's been an extraordinary year for charities globally, not just here in the UK. The industry is concerned that we're going to lose possibly 10% of current charities simply because of the lack of fundraising, etc., a lot of lack of activities ongoing, which is a real concern. CPT has been very lucky. We've had some incredible fundraising highs from the efforts of some amazing supporters. We had the Cure Cubed art exhibition. Our new patron, Paul Mayhew Archer, the comedian and writer, he raised one of the highest totals on the Radio 4 appeal. And then we also had the successful Raid Local, which was a cycle challenge. It was originally supposed to occur across the Swiss Alps and down into Italy, but given COVID, it shifted to a local situation. But that allowed others to get involved, and we had people all over the world getting involved with this cycling fundraiser, and it was very exciting. And we've been very lucky with regards to a lot of our regular supporters, trusts and foundations, major donors. But it has been a hard year for fundraising for CPT, and we won't really know until the end of the year exactly how badly we've been affected, but it has been challenging.
Thank you, Simon. I think Sue now has a question to ask.
Thank you, Simon. I don't feel quite so bad now about not being able to follow all the science that's coming out so quickly. So thank you very much for giving me a clearer conscience. There seems to be a lot of emphasis on drugs designed to combat specific genetic causes of Parkinson's. But how likely is that these drugs could be useful for people with idiopathic Parkinson's?
So what I'm going to do again is share my screen. And I want to show you one other graph that was actually in the presentation, but it's good to have another look at it. So this is from the Ambroxol study. And what you have, as I said, there are people in this study that have a genetic mutation and those who don't have a genetic mutation. So you've got white circles and black circles. And what you notice is that despite the fact that people have got genetic mutation, which results in lower levels of GCase activity, it's a bit of a mix. Both there's black and white dots sort of down the bottom here. There are some individuals who have a normal GBA gene, but they've got very low levels of GCase activity protein in their brains. So it might be that while Ambroxol is focused on improving GBA or GCase levels in the individuals with a genetic mutation, it could be that these individuals with very low levels of GCase could benefit as well. So this is an individual with idiopathic Parkinson's effectively. I think he or she could benefit as well from this treatment. So going forward, we're going to have a situation where personalized medicine is going to have to focus on determining levels of these proteins. That is if Ambroxol is found to be effective. And if somebody has a low level of GCase activity, we could start them on an Ambroxol-like drug and see if it boosts the level of GCase activity, and if that goes on to benefit their Parkinson's. But GCase is not the only protein. Also LRRK2, individuals with idiopathic Parkinson's have been found to have hyperactive or higher activity in their LRRK2 protein as well. So while it could be beneficial for an individual with a LRRK2 mutation, It might also be very helpful to individuals with idiopathic Parkinson's. So that's where a lot of the genetic work is quite exciting, because it does have the potential to flow on to the idiopathic community.
Thank you very much. And I'm going to pass back to Ian now. Thank you, Simon.
Yes, Simon, there's growing evidence that Parkinson's could originate from environmental factors such as pesticides. What is your belief, and is Cure Parkinson's Trust involved in any research into this very contentious area?
We don't know what the cause of Parkinson's is, and it is possible that there are multiple causes. There are certainly a lot of indications that environmental factors like pesticides and air pollution could be influential factors. The Cure Parkinson's Trust, however, is focused solely on disease modification. And while we are very supportive of research that explores causal factors, we are more interested in trying to determine the components that stratify individuals, which will hopefully allow us to conduct cleaner and better clinical trials focused on allowing us to focus specific treatments on specific subtypes of Parkinson's.
Thank you, Simon.
I think I had another question, something I always wanted to ask. Looking at Parkinson's from within, looking at research from within a condition that measures success in number of decades, I can only enviously look at conditions such as the COVID-19, where the expectations are sort of a year to year and a half, and pressure even to go down with that number. Can you explain the main difference between the two areas that drive these changes?
So COVID is fortunate. It's a horrible condition, but it's fortunate in that we know what the causal factor is. And we've had success previously in treating similar conditions. So the causal factor is a virus, and we have lots of antiviral treatments. You could look to AIDS as an example. People with AIDS can live normal life, and their well-being is certainly improved from what it was in the 1980s and 90s. So that is where a condition like COVID is, where we are lucky with a condition like COVID. We know what the causal factor is. With Parkinson's, we don't know what the causal factor is. There's possibly multiple causal factors. And that certainly complicates the picture. I'm pretty optimistic, however. I think within my lifetime, we'll certainly have disease modification. And within my daughter's lifetime, I think we will be knocking it on the head. She's 8, by the way, just to put that in reference. I think there's so much research going on at the moment in so many different areas that we will see positive outcomes in some of these clinical trials. And I don't want to raise expectations there. I'm doing my best to lower everybody's expectations. But there's just so much research going on. And it's overwhelming for a researcher communicator like myself to try and keep up. But one of the just changing tact slightly, based on the COVID nature of the question, one of the benefits of COVID has been the templating of a lot of the efforts, the rapidity with regards to how fast the clinical trials are getting initiated, and the decisions are being made. A lot of that is now being applied to other areas, Alzheimer's, Parkinson's, cancer, and also the structure of the clinical trials. They're using large platforms where multiple arms, multiple treatments are being tested at the same time against a single placebo. We're also looking at that. Camille Carroll down your way is proposing a multi-arm, multi-stage platform for Parkinson's, and that's being investigated. I think it's an exciting time, and it's unfortunate that events like COVID have to come along to sort of initiate and really incentivize us to make the change. But if we can take any positives from the current situation, I think a lot of these changes that will come as a result of it are certainly positive.
All right, I think we're at the end of the pre-prepared question. I'm not sure what your timing is looking like, but is it okay if I open it up to the panel for some additional questions?
As long as they're general, yes.
Anybody has any other questions they want to pose to Simon?
Yes, I do.
Okay, go ahead.
If one of these treatments proves to be useful in slowing down the progression of the disease, in your opinion, at what stage of the disease do you think it would be too late to begin this type of therapy? I speak with concern for myself, really, as well as others in my position. I've had Parkinson's for about 15 years now, and I'm now on the waiting list for DBS, and wondering if there's going to be anything else as a choice.
Well, within the last few years, there's been a recognition of this as a factor in clinical trial design and in future treatment approaches. We have different stages of Parkinson's. For example, we have this pre-diagnosis phase, which is called the prodromal phase. And there are efforts to design clinical trials focused on testing drugs before people are diagnosed. And then there is the de novo stage, which is recently diagnosed individuals. And we've got clinical trials focused on them. For example, the PASADENA study I talked about. And then there is the late stage, individuals in later stages of Parkinson's, and we have clinical trials focused on those as well. There are a lot of different trials being conducted focusing on these different areas. Prodromal is still in development, but de novo and the various stages of diagnosed individuals, clinical trials are being conducted for that. It depends on the nature of the therapy being attempted. So you could take a any sort of curative therapy for Parkinson's is going to focus on three factors. Firstly, stopping or slowing the condition. Secondly, offering some sort of neuroprotective approach to support the remaining cells and provide some sort of nurturing environment for the third factor, which is some sort of cell replacement approach, some sort of restorative approach. And so if you have just been diagnosed, for example, you might not need the last two. You just need a bit of a treatment that'll slow or stop the progression of the condition. And that could come in the form of immunotherapy that we're talking about, this vaccine approach. Whereas for people who are at later stages of Parkinson's, we need to stop the spread of the condition, also have some sort of neuroprotective approach. So a GDNF, which we didn't discuss here today, but a GDNF approach or an Exenatide approach, plus some sort of cell replacement therapy. And all of this is being explored currently in the clinic. Even GDNF, there's a gene therapy clinical trial for GDNF ongoing as we speak. And there's certainly a lot of cell transplantation work. So it's a fascinating time for Parkinson's research in this regard. We're no longer simply throwing a single drug at all sorts of people at all sorts of different stages of the condition. We're really targeting specific subtypes of Parkinson's in that continuum. And ultimately, that's what the treatment will look like longer term. The doctor will be bringing out a menu of different options and designing the treatment around the individual where they are in that continuum.
Thank you. That sounds very interesting and hopeful.
Lower your expectations, though.
Yeah, all the time.
Anybody else has a question?
Yes, I do, if I may, please. Hello, Simon. My name's Nigel Coleman. Another COVID question. Massive amounts of media press at the moment sort of talk about an epidemic of PD in survivors of COVID. Do you think there's any foundation in this information?
Thank you, Nigel. It's a really interesting question, pertinent to the moment. And it stems basically from the 1918 Spanish flu pandemic, where there were individuals after that displayed evidence of Parkinson's-like symptoms. They're now not sure that it was influenza that was causing those particular cases, and they were very rare. More recently, we have 4 cases of Parkinson's in the literature at present that have been diagnosed post-infection with COVID. That is 4 out of the 30 million known cases of COVID. So it's a very, very rare event. Only time is going to really tell with regards to the long-term impact of COVID. Perhaps a viral infection, it could be an influential factor for individuals that are predisposed to developing Parkinson's. As I say, all we can do at the moment is wait. I know that's a horrible thing to say, but we just have to wait and see. It's dangerous to speculate, I think.
Yes, so we come to the end of this Meet the Researchers Hour. Really like to thank Simon for his time and his exciting presentation. I personally feel very encouraged and invigorated by what you've told us. I'd like to thank the PenPRIG team and the volunteers for having put this together, and our supporters who've joined us for this presentation for the first time. As I said, we are a team of volunteers, so if you see in this and you want to get involved in helping us, we're always looking for additional helpers and support, so please look us up on either Facebook or on our website. And please look out for the next event. You'll be pleased to hear we're already looking to schedule the next one, and COVID will hopefully allow us at some point to meet in person, which is what we really want to do, rather than just over the internet. So with that, I say thank you and see you all again.
If everybody would like to unmute and give them our customary Zoom applause.
We have to bang pots and pans.
Bang the table.